Okay, so we are working on those 4 biological molecules, and we have gone over carbohydrates, CH and O, and then we've gone over lipids, very hydrophobic. We have now talked about proteins, super diverse. They have that amino and the carboxyl group coming together to make peptide bonds. And proteins are complicated because they have all those different structures. Primary, the sequence of amino acids. Secondary, the backbone held together by those peptide bonds. Then we have the tertiary structure, which is the 3-dimensional shape. And then we can have a quaternary structure where those two 3-dimensional shapes can come together. So the last group of biological molecules that we have to talk about are nucleic acids. And these hold all of the hereditary information. So they're pretty cool. So let me share my pad. Oh, jeezy. I'm doing something crazy. Oh, no. Oh, hold on. Bear with me. So sorry. This always takes me just a moment to get it together. I usually do, not always. All right. So nucleic acids are the last group of biological molecules that we have to talk about. Remember, all biological molecules have what in common? They have carbon containing, and they're all made of monomers that come together through dehydration synthesis reactions to form polymers. All right, so nucleic acids store, transmit, and help express hereditary information, right? They are made of nucleotides. So nucleotides, and I usually abbreviate that with an NT, the nucleotides are the monomers, right? Those are the monomers for nucleic acids. The nucleic acid is the polymer. And again, just like all of the others, they're formed through dehydration synthesis reactions. The sequence of the sequence of nucleotides. So again, just like proteins, with proteins, we had the sequence of amino acids, depending on what amino acid you had, amino acid 1, 2, 3, 4, that kind of dictated the structure and the function. The similar thing happens with nucleic acids. The sequence of the sequence of nucleotides, nucleotide 1, nucleotide 2, nucleotide 3, nucleotide 4, etc., will determine the hereditary information that we're talking about. And in fact, what we're going to learn about, which is really, really cool, is that the nucleotides actually determine the proteins in our bodies, right? So we get kind of like, I'm going to call it info flow. Information flows from our nucleic acids, and they dictate what our protein structures will be, or protein sequences will be, which is really neat. All right, we have 2 main types of nucleic acids. We have DNA and RNA. DNA is deoxyribonucleic acid, where DNA is just ribonucleic acid. Regardless of what nucleic acid you are made of these nucleotides, these NTs, which all have 3 parts, all right? They have a phosphate group, right? They have a 5-carbon sugar, and then they have a nitrogenous base, or a nitrogen-containing base, right? Those are a nucleotide. So nucleotides, phosphate group, 5-carbon sugar, nitrogenous or nitrogen-containing base. Here's just an example of a nucleic acid that we're going to see a lot. It's called ATP. You don't have to know the full name, adenosine triphosphate. But kind of knowing the name sometimes helps you determine what it would look like. This is an energy carrying nucleotide that has 1, 2, 3 different phosphate groups on it. We're going to talk about ATP a lot because it's really highly involved in energy. However, this is just a nucleotide, right? So this is just a nucleotide, and you can look at it. It has a phosphate group, it has a 5-carbon sugar, and it has a nitrogenous containing base. So again, on a test or a quiz or an assessment, I'm going to show you a molecule that you have never seen before, and you have to identify, is it a carbohydrate, a lipid, a nucleic acid, or a protein? So I'm trying to give you clues onto how to recognize those. Nucleotides always have nitrogenous containing base, 5-carbon sugar, and a phosphate group. All right, when you look at a nucleotide, and again, the 3 parts, it has 1 is a phosphate group, 2, it has a 5-carbon sugar, and 3, it has a nitrogenous base. If you look at the sugar, it comes in 2 forms, and that dictates what kind of nucleic acid it will be. If it has a deoxyribose sugar, then it will be deoxyribonucleic acid, or DNA. And if it has a ribose sugar, that will tell you that it is RNA, right? Or ribonucleic acid. And if you look at the 5' sugar, there's an easy way to tell whether it's deoxyribose or ribose. And it happens to be right in the name, deoxyribose. So if you notice here, usually you have a hydroxyl group. However, with the deoxy, you're missing one of those oxygens, right? So DNA and RNA polymers, or nucleic acids, differ in this 5-carbon sugar. What we're going to see is that the DNA on our chromosomes in the nucleus of our cells is DNA, right? But RNA is going to be something that is used to make copies of that DNA that can never leave the cell, right? We'll go over all of that eventually, but this is all about kind of information flowing from the DNA to the RNA, and the RNA is going to help make proteins, which now you know a little bit about. All right, so let's look more carefully at DNA and RNA. Sorry, again, the formatting is a little funny, but on the PowerPoint that's posted, should be all good. All right, so some of you probably already know this, but I'm going to start over here with the red and blue ones. So most of you already know that DNA is a double helix, right? So it means 2 strands of DNA wrapping around each other. RNA, you might not know about, is single-stranded. So there's another difference. Not only do they have differences in their sugars, but also in their full structures. Okay, if you look at the structure of, let's say, DNA at the beginning here, the back, well, DNA and RNA, they all have what's called a, I'm gonna write this up, a sugar phosphate backbone, right? Meaning this part in blue is always going to go sugar, phosphate, sugar, phosphate, sugar, phosphate, etc. Right? Remember, every nucleotide is going to have a single phosphate group, a 5-carbon sugar, and a nitrogenous base. But the backbone is always going to be sugar, phosphate, sugar, phosphate. And they come together just through a dehydration synthesis, but they form a bond that's called a phosphodiester bond between the sugar and the phosphate groups. I'll write that down. Phosphodiester bond, right? So again, carbohydrates come together through glycosidic bonds. Proteins come together through peptide bonds, and DNA and RNA are held together by phosphodiester bonds on that backbone. And one other interesting thing is that phosphate groups, you already know this from your functional groups, is that they're highly negative, right? So the backbone of DNA and RNA is negatively charged, which is going to be useful when we talk about, when we go into our biological techniques on how to deal with DNA and RNA, all right? So there's lots of let me go on. We're not going to talk too much right now about the different kinds of nitrogenous bases, but you may already know that DNA has, for example, A, T, C, and G. May know this, may not. And RNA has the nitrogenous bases A, U, C, D, right? And don't worry about this if you haven't I'm never gonna make you know the structure of these ever. You'd never have to recognize them or anything. But what you do have to know is that G's and C's come together and A's and T's come together. And the way they come together is through hydrogen bonding. Remember, hydrogen bonding can be between O's and H's or H's and N's, right? And remember, these are nitrogenous bases. So these are the N bases that we're talking about. And these nitrogenous bases has a lot of nitrogen. And we know that hydrogen bonding is between N's and H's. All right, so if we look at the structure of just DNA, it is in two strands of DNA come together in a form which we call anti-parallel. They're side by side, like railroad tracks, but they're pointed in opposite directions, right? They're pointed in opposite directions. So for example, if we look at that backbone, sugar, phosphate, this direction, we see that there is a sugar at the end, right? Sugar at the end. Here, on this side of the DNA, we notice that there is a phosphate at the end. So they come together in opposite directions, but they're bound together through hydrogen bonding of these nitrogenous bases. And we're gonna go over a lot more of the structure of DNA when we get to that. All right, so RNA is like DNA, but it's single-stranded, so it doesn't have those hydrogen bonding. And DNA, here, let me actually just go here. So the main points about nucleic acids, they're made of nucleotides, and the nucleotides have 1 phosphate group, 2, they have a 5-carbon sugar, and those 5 carbon sugars can either be deoxyribose, or they can have a ribose sugar. Remember, deoxyribose, instead of having an OH, it has an H in one of the positions. And the ribose has an OH. And the third is that they have nitrogenous bases, right? So these are the monomers. And they come together to form phosphodiester bonds. Those form between the sugar and the phosphate group, and that forms kind of the backbone of all nucleic acids. So if we had, for example, we could have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, etc. And if they came together, for example, in the form of DNA, these sugar, phosphate, would, the nitrogenous base here would come together. So these nitrogenous bases would come together through hydrogen bonds. And remember, hydrogen bonds are very weak individually, but collectively they're very strong. In DNA, when you have DNA come together through hydrogen bonding, there's many, many, many of them, making them fairly strong. All right, and we'll stop there for nucleic acids.